Why split a trial into small incomplete blocks at all? It comes down to one trade-off. Picture a field with a fertility trend running across it. If a whole replication is one big block, plots at opposite ends sit on very different soil, so the within-block error variance is large, and real variety differences are hard to see. Break it into small blocks and each block covers nearly uniform soil. The within-block variance drops, and comparisons within a block get sharper. But there is a catch. Varieties in different blocks are not compared directly, so some information is lost. Precision depends on two things: the within-block variance sigma squared, and the efficiency, E. The block size sets the within-block variance, and that is the experimenter's choice. The efficiency factor, E, between 0 and 1, is the fraction of comparison precision you keep despite the blocking. The design maximizes that efficiency in just one way. By minimizing co-occurrences, keeping any two varieties from sharing a block more often than they must. That one goal drives everything that follows. Suppose we want to compare 20 varieties in a field trial, with 3 full replications. A whole replication in one long strip is noisy, because distant plots differ in soil and moisture. So we break each replication into smaller, more uniform and complete blocks, 4 blocks of 5 plots each. 3 replications in all, and 20 varieties, since 20 is 5 times 4. The challenge is choosing which varieties share a block, so that comparisons stay balanced. Patterson and Williams gave a simple recipe. The recipe starts with a generating array, 5 rows for the 5 plots in a block, 3 columns for the 3 replications. Every entry is a number from 0 to 3, a residue modulo 4, where 4 is the number of blocks in a replication. This array is in reduced form. The whole first row and first column are 0. These few numbers are the entire seed of the design. And these numbers are not arbitrary. This little array is pre-computed, looked up from tables, given by a formula, or found by search, and chosen so that the finished design maximizes the efficiency factor, E. Picking the array is the only clever step. Everything after it is mechanical. Now we develop each column cyclically. First, the 3 columns of the generating array slide apart, to make room. Take each column and make 3 more copies, adding 1 to every number each time, and whenever a 3 would become 4, it wraps back around to 0. Each original column blossoms into a group of 4. The 3 columns of the seed become 12 columns in this intermediate array. So far every number is small, between 0 and 3. To turn them into variety labels from 0 to 19, we shift each row by a fixed amount. The first row stays. We add 4 to the second row, 8 to the third, 12 to the fourth, and 16 to the fifth. Now all 20 varieties appear, and each one shows up exactly once in every group of 4 columns. These 12 columns are the blocks of the design. First, watch them separate at the replication boundaries. Replication 1. Replication 2. Replication 3. Each group of 4 columns is one complete replication, containing all 20 varieties. So the design is resolvable. And each column is a block, a set of 5 varieties grown together. Look at varieties 4 and 16. They share a block twice, once in replication 1 and once in replication 3. And notice they are not neighbors, other plots sit between them in the column. Co-occurring means sharing a block, not being side by side. Most pairs meet 0 or 1 times, a few meet twice. That tight co-occurrence structure makes the design efficient. This is an alpha 0, 1, 2 design. Here is the whole co-occurrence structure as a heat map. Each cell is a pair of varieties, blue if they never share a block, green if they share 1, red if they share 2. The gray diagonal is a variety with itself. Counting them, 82 pairs never meet, 96 meet once, and 12 meet twice. Those 12 red cells are what make this an alpha 0, 1, 2 design. The plan is systematic, so before planting we randomize it. But watch varieties 4 and 16, which share a block twice, and note they are never side by side, other plots sit between them. The maroon boxes will follow them through every shuffle. First we shuffle the order of the 3 replications. Whole replications switch places, the numbers inside each one stay exactly as they were. Then, within each replication, we shuffle the order of the blocks. 2 boxes still, 2 shared blocks still. Within each block, we shuffle the plot positions. 4 and 16 slide around inside their blocks, the gap between them even changes, but they stay in the same block. Proximity is not the same as sharing a block. Finally we relabel every variety. 4 becomes 19, and 16 becomes 15. The boxes do not move, because it is the very same 2 blocks. Only the names changed. The co-occurrence count never changed, still exactly 2. Randomization preserves the design's co-occurrence structure, while spreading any field trend randomly across varieties. A fair, efficient, ready-to-plant alpha-lattice layout. Part 2 is a different design, a separate trial, with 30 varieties, illustrating the improved lattice you get when the block size K is at most the number of blocks S. Here, blocks of 5 plots and 6 blocks per replication, K equals 5, S equals 6. 3 replications, 30 varieties in all, since 30 is 5 times 6. Because K is at most S, no pair is ever forced to share a block more than once, the improvement of an alpha-01 design over an alpha-012. How is that design built? For this case, 3 replications and an even number of blocks, the paper gives a Series 3 construction, the same family field hub uses. Here is its generating array, 5 rows, 1 per plot and a block, and 3 columns, 1 per replication. Developing it modulo 6, and shifting the rows by 0, 5, 10, 15 and 20, produces exactly the 30 variety design we just saw. Track varieties 0 and 12. They share exactly 1 block, here, and they never appear together anywhere else. And notice they are not neighbors, another plot sits between them and the block. Co-occurring is about sharing a block, not about being next to each other. Across all 435 pairs, 255 never meet, 180 meet in exactly 1 block, and 0 meet twice. No pair is forced to co-occur twice, because the block size does not exceed the number of blocks. That is an alpha-01 design. Here is the co-occurrence heat map for this alpha-01 design. 30 varieties, blue and green only. There is no red, because no pair shares 2 blocks. Of the 435 pairs, 255 never meet, 180 meet once, and none meet twice. This scales straight to your real trial. 50 entries, blocks of 5, 10 blocks per replication, 3 replications, generated by Field Hub with the same Series 3 construction. Because the block size, 5, is at most the number of blocks, 10, it is an alpha-01 design. Of all 1,225 pairs, 925 never meet, 300 meet exactly once, and not 1 pair is forced to co-occur twice. Same recipe, same randomization, larger field. Construct once, randomize per site. The same heat map for your Field Hub design. 50 entries, so 2,500 cells, and only blue and green. There is no red anywhere. 925 pairs never meet, 300 meet once, and not a single pair meets twice. A clean alpha-01 design.